Hello, everyone. In this presentation, I would like to introduce to you our TARGATT technology, which is a rapid, site-specific gene integration technology for making stable cell lines for bioproduction, as well as for mammalian library screening. TARGATT technology allows site-specific irreversible gene insertion and works in non-dividing cells and allows large fragment DNA insertion up to 20 kb. Some of the key features of a TARGATT includes controlled copy number insertion and has high integration efficiency up to 40%. And the expression of the transgene is consistent across constructs. This is because these transgenes are inserted in well-defined safe harbor genomic locus. And this allows protein expression from medium to high level. And the cell lines made with TARGATT is also stable with stable gene integration. So we have generated TARGATT Master Cell Lines that contain the landing pad that is recognized by TARGATT enzyme. So these pads here are designated by the purple sites are ATT or attachment sites, and TARGATT enzyme recombines between these ATTP sites, the attB on the vector, and results in site-specific integration to gene. And we also engineered suicidal genes. We have generated TARGATT master cell line that contains TARGATT enzyme recognition site, so-called docking site or attachment sites. ATTP, we have generated TARGATT master cell line. In these cell lines, we have engineered TARGATT enzyme recognition sites in a safe harbor genomic locus. For example, in this case, we have engineered ATTP sites which are recognized by TARGATT enzyme into a safe harbor locus in the genome. When an incoming vector that contains the ATTB site together with the gene of interest, recombination between these sites results in site-specific integration of either the gene of interest or the other site which contains a TK gene. And by adding ganciclovir, it kills all the integration that contains the TK gene, so results in the clones that only contains the gene of interest. So this is just to show that TARGATT CHO master cell line allows high efficiency stable cell line generation. So the top two panels are images from control cells, which has random integration, and the bottom two contains TARGATT integration cell lines. As you can see, we're able to achieve over 97% of insertion after drug selection. And we also confirmed that the integration of the transgene is single copy by using color labeled constructs. And we have identified several genomic safe harbor loci to engineer various landing pad into the genome. So the H1 locus is the locus we use as a benchmark. And this particular locus has been proven to be a safe harbor genomic locus with high gene expression in many different systems. For example, in the mouse, in the pigs, in CHO cells, and also in human iPS cell and ES cells. And this just to show that in the CHO cells, we're able to see good expression at H11 locus. And this is to look at many other sites. So these are new sites that we identified that has good expression as well as genomic safe harbor loci. So as you can see, so on the left graph is a expression using GFP over a period of time. So these are from 21 days, 42 days, and 82-day cells. And as you can see that ASC2, this particular locus, allows the highest expression. And then on the right side, so these cells were analyzed at the 56 days post-transfection. And again, just to confirm that ASC2 performed very well among all the other sites that we identified. And now we use a more relevant protein. So this is to look at expression of Fc fusion protein. And this is just the process of making these cell lines. And this is the analysis of protein expression, titers from 6-day Shake Flask in the lab. And so this is ASC2 sites in CHO cells. And these cell lines has the H11 locus. And as you can see that ASC2 performed even better than H11 in terms of protein expression. And so, and also there's like, these are different number of clones. And there's a good consistency in terms of protein expression across the clones. And this is another test that we did in expression of antibodies. So there are two antibodies, antibody 1 and 2. And experiment was done in a 17-day fed batch Shake Flask. And we were able to see about 1 gram per liter antibody production in this particular target CHO cell line using the H11 locus. So this is just to compare our target technology with random insertion, which is the current technology used for generating CHO cells, and some of the newer technologies, zinc finger nucleus, TALEN, or CRISPR technology. So as I mentioned before, so target allows site-specific gene insertion, and the big advantage of target is really its efficiency. So we have been able to get very high efficiency, up to 40%, and also we're able to insert large fragment up to 20 kb. So now I'd like to talk about using target for screening libraries in mammalian cells. So currently, most library screening are done in bacteria or yeast. However, as you can see that these systems lack some of the translational, post-translational modifications. And the mammalian system is really a better reflection of expressing mammalian proteins. However, the current available systems are slow and very labor intensive. So we're using our target technology to establish a screening system. So because our efficiency, we're able to build a library that allows very high coverage of the diversity of the library. So this is just to compare the current available technology and their coverage of the library size. So bacteria and yeast are the most used systems for making libraries because they are high coverage. So bacteria system, for example, can cover up to 10 to the 9th coverage. And then these black bars are actually a more recent development on using mammalian system for library screening. And their coverage is relatively low or much lower in this case compared to libraries using yeast or bacteria. So our goal is to really using target technology and to consistently hit within an order of magnitude that is typical for bacteria or yeast libraries. So the way we do this is that we use our target technology and a split landing pad, a split cassette selection system. And so the landing pad that for the target technology is inserted in the genome. And then in the donor DNA, we have the attB site. So, and mCherry is not expressed in the vector because it does not have a promoter. And then the promoter EF1 is in the genome. So upon recombination between these attP and attB sites results in integration of mCherry and brings in mCherry to EF1 alpha promoter, allows expression of this red fluorescence marker. So that's how we screen for positive clones. And so we're able to reach, using this system, we're able to reach up to 40% integration efficiency without drug selection. And with drug selection, we're able to reach over 90%. So this is really the highest efficiency that has been reported in mammalian cells. And so this is just to project what kind of coverage of library size we will be able to get using target system compared to some of the other reported library screening in mammalian systems. So basically, this is just bacteria and yeast as reference. And so these other methods reported various coverage. And so our target DNA integration efficiency is 1,600 to 4,000-fold improvement over other systems, right? Flip in. So most of these systems using flip in system. So it's flip recombinase system. And so basically it's 40% versus 0.01% to 0.025%. And so we will be able to have much better coverage and by screening less size of clones. So basically, I hope I have explained to you about how to use our target technology for generating cell lines for bioproduction and also for library screening. And we have a poster that specifically describe how we use target technology for library screening. So please go to our poster and take a look at that work. And thank you very much.